Question mark. So like, I'm curious right now, is it recording what I'm sharing on the screen or my whole computer screen? You know what I'm saying? Guess we're gonna find out. We'll just do it and we'll just see what happens. Hello. I'm signed in. How do I find, oh, they always change the layout of this. Okay. It's recording the chat. I don't know. I'm sorry. Wow. All right. Let's get serious here. Sign up for Kahoot. Pee pee poo. Really? All right, anyone else that wants me to wait before we get started? I think we're good, right? Because Brian said his internet is poop. All righty. Which of the following is not true about meiosis? Good. So it is cell division for gamete cells or sex cells. It begins with 4 cells, begins with 1 cell, begins with 4, ends with 4. Begins 4 and ends N1. And there are 2 divisions. Freaking bubble chat. I feel like that's Eli. What is the proper term for sex cells? Gamete. Good. That one right there is Miles. It happens to all the good ones. It's okay. Which of the following is not a gamete? Good. Skin, right? Sperm and egg cells are sex cells. You guys are crazy. Bingus. Bingus has a streak. Bingus. Oh my God. How much genetic info do cells begin with in meiosis? Good. 4N. O. N is the quantity of genetic information. How much genetic info do cells end up with in meiosis? If what you're asking, Fadl, is, are there other options than 4N, 2N, or 1N? Yes, in different organisms. And different organisms also have different quantities of chromosomes than humans. And it's not relative to how big or complex the organism is. So some smaller organisms have a lot more chromosomes than we do, and a lot lengthier amount of DNA. Some larger organisms have a lot less than us. Do I know how long the front office is open today? Because you need to finish your work permit. So they normally stay for the full length of a teacher work day, which goes until about 3:20. So typically about 3:30, 4 o'clock they're open till. Bubble Chat has new streak. The cells produced during meiosis are genetically unique. What is synapsis? Good. So pairing up of homologous chromosomes. What are homologous chromosomes? Pairs of sister chromatids with unique alleles. What's an allele? Variation of a gene. Define chiasmata. All right, good. When the homologous chromosomes overlap, or as Valentina said, they hug. What occurs during crossing over? Homologous chromosomes swap alleles. Oh, meow in the lead. Your names. Which of the following does not make sense with prophase 1? Good, metaphase plate. So the synapses and chiasmata form, crossing over does occur. Metaphase plate happens shockingly in metaphase. What is the metaphase plate? Good, the center of the cell. What type of chromosomes are involved in meiosis 1? Homologous pairs. Okay, Paulette, what do spindle fibers do? Move chromosomes, good. Luffy for the lead. What type of chromosomes are involved in meiosis 2? Good, sister chromatids. Remember, homologous chromosomes were in meiosis 1. What does the law of independent assortment refer to? Chromatids separating randomly of each other. So this happens again in anaphase 2 of meiosis. And again, it guarantees that the variations go randomly into sex cells. So they don't all clump together based on dominant or recessive. They don't all clump together based on like they came from mom or they came from dad. They're just all mixed up. And that mixing up and random separating that happens then in anaphase 2, in addition to crossing over, is what helps to increase the genetic variation of the sex cells. In general, what happens during metaphase? Stuff lines up. What about what happens during anaphase? Stuff separates. Fadal is in love with metaphase. What happens during telophase? stuff finishes separating, right? And so when we're saying stuff right, this is me thinking back to mitosis and meiosis 1 and meiosis 2. It's either sister chromatids we're talking about, individual chromosomes or homologous chromosomes, but the same basic movement or event happens in all of those. All right, what about cytokinesis? Right, good, the cells actually divide. And then lastly, how many cells do we end up with in meiosis? Oh, sorry. sorry. All right, good. Okay, let's see who our big winners are here. All right, Luffy, Bubble Chat, and Meow. All right. Your names. So if we can go into Canvas. Modules. So here you can see your 5.3 practice. This assignment, your usual vocab, diagrams, and concept application, that is going to be due this Sunday night. I'm going to go over now just to start to introduce it. Your kind of like lab project activity, I don't know what you want to call it, okay? It doesn't really matter what we call it because everything is graded equally. But this is something that I want to introduce now because I'm sure there are some of you in here who will start thinking about it this weekend. Again, it'll be due a week from Sunday. You will have both class periods next week to work on it. It is definitely something that I would imagine, if I open up the link, I can estimate for you how long I think it would take you. Okay. So it says here, let's apply what we've learned about chromosomes and meiosis to create and evaluate a karyotype. In this lab, we are going to learn about karyotypes, how to analyze karyotypes, and assign chromosomal disorder and how chromosomal disorders originate during meiosis. Click on the assignment link below to open it up, read about it, and create your karyotype. Complete a karyotype and model. Directions are in the assignment. There are 4 sets of chromosomes. Now I have listed here the chromosome set you are assigned. Notice it says, make sure to make a copy first. Do we all see that? Make a copy first. Okay, I'm just saying, someone in here, if someone doesn't make a copy, you're gonna really mess it up for everyone else, okay? So right now, if you could click on your assigned set, I just happened to click on number 1. To make a copy, what you're gonna do is go to File, click on make a copy, and then rename it. Okay, so I could be like Fadl's chromosome set number 1, Rosema's chromosome set number 1, okay? And now you have your own chromosome set that will be saved to your drive, and that's the set that you always want to refer back to for this project. So again, here is the list of the chromosome sets you are assigned to. Click on the one you're assigned to. When you open it, click on File and make a copy. And then rename it so it has your name in the chromosome set. And then that's the one you're always going to use for this project. Okay, is that working for everyone right now to make a copy? Does anyone need help? Okay, was everyone here able to make a copy? Okay, I will assume that means yes for the rest of everyone. All right, so now let's click on the assignment. So if I just take a second to scan through this assignment, we've got a good amount of background information, okay, and karyotypes, which are going to be helpful for you. Then in part 3, I have directions, and I'm going to go over all of this. And part 4 is your actual like project you make or create, okay? So up at the top here, it says, background information. Karyotyping is a process that scientists and geneticists use to sort and look at an individual's chromosomes and identify possible chromosomal abnormalities or disorders that could arise. The individual's chromosomes are stained so that their unique characteristics are more visible and arranged by homologous pairing, length, long to short, and then the individual's biological sex chromosomes. Humans should have 46 total chromosomes or 23 pairs and should have 2 sex chromosomes, either 2 X chromosomes or an X and a Y. The following are common abnormalities that scientists or doctors would look for. So chromosomes that are missing chunks or ends, and they call that a deletion. Chromosomes having extra chunks called a duplication. A chunk of one chromosome is transferred to another chromosome called a translocation. A chunk of one chromosome is removed, flipped upside down, and reinserted, called an inversion. You have too many chromosomes called trisomy, or you're missing chromosomes, monosomy. And when I refer to chunks, remember that chromosomes are just wound up DNA. So when I say like a chunk is removed or missing or added, we're saying a sequence of DNA is removed, added, flipped upside down, etc. Okay. Now, the ones we're going to be focusing on here in this project will be trisomy and monosomy. So then it says chromosome abnormalities usually occur when there's an error in cell division. There are 2 kinds of cell division, mitosis and meiosis. And we just learned about those, so I'm not going to read over that. In both processes, the correct number of chromosomes is supposed to end up in resulting cells. However, errors in cell division where chromosomes are not properly lined up and split up can result in cells with too few or too many copies of a chromosome. Other factors can increase the risk of chromosome abnormalities. So maternal age. Remember we talked about there being more errors in female eggs the older they become. Environmental issues could be chemicals, that are in foods, pesticides, things like that. But also, unfortunately, it just sometimes happens. So let's take a look at some karyotypes here. This is a karyotype, and these little gray, white, and black kind of striped chunks are chromosomes or wound up DNA. When I look at this karyotype, first I can see, I mean, it's a little bit faint, and you'll be able to see better in your actual chromosomes you're working with. There is a unique striping or banding pattern in each of these sets of chromosomes. I can see that the pairs of chromosomes match up by length, by shape, and by banding pattern. I can see that pair number 1 here in the top left corner is the longest pair. And then as I move left to right down the rows here, they get shorter and shorter, and pair number 22 is the smallest set of chromosomes. I can see that the sex chromosomes are always in the bottom right corner. This individual has 2 X chromosomes, no Y chromosome, and so therefore is biologically female. I can see that each set of chromosomes has exactly 2, not 1 or 3. Okay. So let's look at another karyotype here. Again, I see the same types of patterns and arrangement. What's different about this karyotype from the last one? It's in the bottom. What's different from this karyotype compared to the last one? Good, this one has 1 X chromosome and 1 Y chromosome. So we would say it's biologically male. And again, though, things I'm looking for, right? I see that each one has the same pair of chromosomes. So I don't see one that has like 3 in a place or one that only has 1, okay? All right, now let's look at this karyotype. Where do we notice something odd in this karyotype? Number 21 at the bottom. So do you see at 21 here, I've got 1, 2, 3 little chromosome chunks instead of just the 2 I see everywhere else. So that's when I have too many chromosomes in a spot. And again, that's called trisomy, when I have too many. I also notice this person has 1 X chromosome and 1 Y, so again, biologically would be considered male. The male chromosome is the smallest chromosome in the human genome. It really only has genetic information for male reproductive parts, and that's it. So having it and not having it is what determines male reproductive parts. Just that one little nugget of DNA. The X chromosome does have a significant amount of genes on it that are necessary beyond reproduction. All right, so now let's look at these directions. Now, I typed out these directions in the order that I think you should do it, okay? It would be a bad idea to go out of order from my list, okay? I promise you. So it first says, you must sort your assigned chromosomes properly into a karyotype. So again, here were my chromosomes. In all of these, I have to pair them together. Longest to shortest, very good, Nicole. In my karyotype table, so here's my karyotype table. You can resize these boxes however you need to, okay? And this is why I wanted you to make your own copy, okay? So as I'm working through here and I'm trying to pair together chromosomes, it is easiest, in my opinion, to just like cut, Control X and paste it wherever it needs to go. Now, I'm not saying that goes there, okay? But that's how I think is the easiest to do that. If you need to turn some chromosomes, remember if you just click on it, it's an image. You can always rotate it, okay? But now that you have your own copy of the chromosomes, you can cut and remove them from the chromosome set and paste them into the karyotype here and try to pair them up and then rearrange them by long to short, okay? And whatever process you do for that, that's up to you, okay? But that way, if you cut it from the original set and paste it here, you guarantee that what's in your original set are only the ones you're still trying to figure out. Again, like Nicole said, it goes longest to shortest. So longest, getting a little bit shorter, getting a little bit shorter left to right, getting a little bit shorter, and then the sex chromosomes, okay? So once you've finished your karyotype, the karyotype part and the model are going to take you the longest. The karyotype part I'm trying to think of the range of humans in this class. I'll be honest, there may be a couple of you that can finish the karyotype in an hour. I would say though probably most of you, if you're focused, it'll take about 2 to 4 hours for you to just do the karyotype. Okay, so start thinking about that and planning ahead. That means I don't want to just work on the karyotype only in my 2 class periods next week. Okay, don't do that. Once I make my karyotype, I am then going to identify the biological sex of the individual, any abnormalities in the individual's karyotype. I am going to use the internets to figure out the disorder that's associated with that abnormality and then research it. So I have my karyotype again. I'm using the X and Y chromosome boxes to figure out if it's biologically male or female. And then I'm looking for boxes that either instead of having 2 sets, have 1 or maybe have 3. Once my karyotype is done, I then fill in this table here. So biological sex, abnormalities. I use the internet to figure out what disorder that is based on where the abnormality is, symptoms of the disorder, and any treatment for the disorder, okay? This part right here, I think is the easiest. Maximum should take an hour, okay? So again, let's think, to ourselves, the karyotype here will take an hour if you typically finish things very quickly and easily in this class. There are some of you that applies to. If you do not, you're looking at 2 to 4 hours to finish the karyotype. I would say maximum another hour to do this research, maximum, okay? And then your meiosis model. So let's go back up to the directions and read what you have to do for the meiosis model. Create a model of meiosis that shows how your assigned chromosomal disorder arose in the individual. Your model, my internet connection is unstable. Okay. Your model should show and explain. And it says, you may hand draw your model, take a picture and insert it below, like you have for graphs. Or you can create a digital model using Google Jamboard or Draw, take a screenshot and insert it below. So your model has to show all of the phases of meiosis, what happens with genetic information along the way, so what happens with the chromosomes, specific to your assigned disorder. So that means if in your assigned disorder, your person inherits too many chromosomes at place number 10, I need to make sure that I show that number 10 chromosome does not line up and separate properly, okay? What happens with the gamete and its genetic information during prophase 1 and crossing over, during metaphase 1, anaphase 1, telophase 1, cytokinesis 1, and then prophase 2, metaphase 2, anaphase 2, including independent assortment, and telophase 2 and cytokinesis 2, okay? All structures and processes should be labeled Use captions as needed to explain what's happening visually in the model. Use numbers to show a sequence that things happen in. So like this happens first, then second, third, and fourth. You can use arrows to show something moving around, and different colors to represent different structures or types of substances. Now, I'll tell you right now, there is a plethora of diagrams in your notes and on the internet for meiosis. You have to make sure, though, that it is going to show the chromosomes. It has to show alleles because I have to include crossing over, right? Which shows the swapping of alleles. So my chromosomes have to have alleles on them. And that was shown in your notes. And it has to show that whatever place I have a chromosomal abnormality at, it has to show that number chromosome not lining up and separating properly, okay? So Nicole said, for example, if the disorder is a deletion, we look up the disorders related to deletions. So I'll tell you right now, the disorders you're looking at are either going to be trisomy or monosomy, okay? Meaning that you either have one too many sets of chromosomes in a place or you're missing a set in a place. Okay, so for instance, let's say I had one too many chromosomes in place number 1. I would just Google like trisomy chromosome 1. Okay, stuff will come up. Okay, let's say I'm missing a chromosome at place 5. I would type in like monosomy chromosome 5. Okay. Oh, Brian. So, to review how long I think this is going to take, a couple of you karyotype about an hour to 2, most of you 2 to 4 hours. An additional hour here for the research. The models are a big task. I would assume for most of us, our models are going to take anywhere from 2 to 6 hours, okay? So if I were to kind of put up here then, and I can email this to us. And what I'm gonna do right now is kind of what I do for everything that I do every week, okay? So for those of you that are having difficulty figuring out how to balance everything and how much time to set aside to do stuff, this is literally what I do in my planner in a list for every single thing that I have to do every week. And that probably seems bananas for you. But that is how I stay on top of. I have a lot of stuff I have to stay on top of, and that's the only way I can do it. So for my time estimate, I said 2 to 4 hours for the karyotype. I said about 1 hour for the research. And then somewhere between 2 to 6 hours for the model. Now, right now, if you know you take longer to do things, which is not bad, okay? Don't even think it's gonna take me 2 hours to do the karyotype or model. Estimate right now that you're gonna need the 4 and 6 hours, okay? So for my pacing goal, then I might say, try to start the karyotype before Tuesday and finish by Tuesday night, okay? Then I would complete the research Wednesday. I would try to start the model before Thursday, even if it's just a brainstorm. You're like rough sketch outlining what needs to go in your model, okay? It's by nowhere near what you're gonna turn in, but it kind of just sets you up of like, okay, I need to include like this and this and this and this, okay? And then, and work on in class Thursday. That way you can ask questions. So that's why I'm saying start brainstorming it before class Thursday. That way when you come Thursday and you're working on it, you already know what types of things you aren't sure about, you're a little confused about, so you can ask me questions. Okay. If you need extra help, ooh, we don't have school next Friday. We can set up a Zoom time. What's a Zoom time next Friday that you guys could all attend? What's a time that you're all awake? Okay, so like if I said 12 to 2, is that okay? Okay. 12 to 2 p.m. Zoom help. Eli, 2 p.m. is in the afternoon. You'll be busy at 2 p.m. Midwinter break. You have no school next Friday and the following Monday. So the 12 to 2 isn't like I'm going to be direct instructing. It's just a time that you could drop in. Since we technically have no school, it's actually no school for teachers as well. So I don't have to be in Zoom on Friday. But I want to make sure that like if you guys have questions, you can ask me. Okay. So you could drop in at any point in that 12 to 2. You can also always just email me. Okay. So this is kind of right here how I would plan ahead for this. I would first chunk the project. I would never be like, oh, this project will take me 2 nights. No, go chunk by chunk and figure out how much time you estimate it will take you. Again, if you know you take a little bit more time to do things, estimate on the larger end of things. Don't try to be optimistic and be like, oh, that should only take me 2. No, it's better to give yourself more time than less. Then once I have that kind of broken apart, then I look at my schedule next week and I start thinking about, so how should I work on this every day so that it's not Friday night, 2 nights before it's due, and I'm having a life crisis meltdown. I'm crying and swearing at my Chromebook and hating life, okay? So if you do it in chunks leading up, then we don't have to have the complete life meltdown, crying, swearing, anger issues, okay? I mean, you might still, but maybe hopefully less, okay? So that's how I would pace this, okay? So your priority should be first, work on and finish the practice. I made the key for it and I did not, personally, I feel like you've had a lot more difficult College Board questions. So I think it's not too bad. That's due Sunday night. Then Monday, I would start getting on this project. Okay, of course, if you get a head schedule here of what I have paced, awesome. It's better to be ahead than falling behind. Okay. Okay, does anyone need or want to ask any questions about anything? Okay, looks like no. Oh. Ah, very good question, Angel. Yes. So one of the extra chromosomes is either coming from mom or dad, right? So she said, if one of the extra chromosomes came from mom's egg, if one of the eggs has an extra chromosome, does that mean another one of the mom's eggs is missing a chromosome? Yes. So most chromosomal disorders are trisomy or monosomy, which means it's when we have a whole entirely extra chromosome or are missing a chromosome. And in those instances, like Angel's saying, 100%, that means if the one we're dealing with had an extra chromosome, there is another sperm or egg cell laying around that is missing one. You know, Brian, since I got Invisalign, I just feel like the dentist is just like a routine errand I have to run. But actually, I found out I get my Invisalign off in 2 weeks. So that's pretty exciting. Oh no. For wisdom teeth or something else? Yeah. Are you getting all of them out at once? It's better to do it that way. It's always better to get your wisdom teeth out earlier than later and all at once. So ideally, you don't want to wait until your wisdom teeth come in because it can mess up the rest of your teeth. You know, when they are present and they see them in an x-ray, you yank them out. Mm-hmm. Yeah, I didn't have any issues getting my wisdom teeth out. The only thing was my face got enormously puffy, but it got like enormously puffy like 5 days after, like the first 5 days after surgery, I was golden. And I really thought like, nothing's gonna happen to me. I'm amazing. And then 5 days later, I looked like a chipmunk with elephantitis. So I don't. But lots of ice cream, pudding, rice, soft foods. I love the soft foods. Yeah, you know, it's a little bit weird because I had braces when I was like your young fetus ages. So I know my teeth didn't need that much correcting, but I am excited. I can tell, you know, it's small things. I can tell already, like when I'm talking in class and stuff, that I smile more with my teeth showing, which I don't normally. So it, you know, little things like that. Sometimes the little things that only you notice really affect your confidence. New AP testing dates. This email just came. All right. So you are going to have an option to take the AP exam fully digitally, it looks like, which I would prefer because say, you know, disease, you know, transmission. Also, I would assume for a lot of us, you can type faster than you can write. Quest Bridge. What the heck is Quest Bridge? Mr. Hughes said to apply for it. So it kind of looks like maybe like a scholarship housing company or site, right? So it's okay. So it shows 45 different colleges connect through Quest Bridge. And so maybe it kind of looks to me like you apply here and you can get a variety of scholarships based on these 45 schools that are connected to it. Do I think digital will be harder? No. I think they're both going to be equally difficult. I'm so sorry. I would agree. So that's my only thing is that, when we get free response questions, those of you that are getting 3s and 4s on the free response parts of your practice, you type a lot. Like you're typing like full paragraphs. It takes me longer to write than it does to type. I'm also a lot more likely writing to not know that I have grammar or spelling errors. So yeah, so for a class that involves a lot of math, so like AP Chem, AP Calc, if it's taken digitally, it's difficult because you would have to hand write out a lot of the work for the free response. Because in those cases, it's more difficult to type that. For this class, the majority of the free response are just pure text. So you would likely just open up a Google Doc, type it in there, and upload it into the question, which is what they had kids do last year. But that's interesting because then if it pushes it back another couple weeks, that gives us a very nice chunk of time to prep. Obviously, everything we're doing in class right now is prepping for the exam. But my plan was already to spend the 2 weeks leading up to the exam just drilling practice exams. So that will be nice. Are grades posted yet? Grades were posted by teachers. You're welcome to check if you can see them. I don't know. I feel like that's information they don't tell us. But you could check. They would be in MyStar. Oh my God, not till Monday or the weekend. What, Fadl? That seems like forever. Wow. The shadiness of the inconsistency of the information. All right. So again, we play Kahoot. You have practice. Okay, your typical practice due Sunday night. I would then recommend Monday you start working on the karyotyping project. I'm going to email you my pacing in case it helps any of you, and if it helps none of you, then that's fine. You'll have next week to work on the project. It'll be due a week from Sunday. Okay. If you have questions, email me, of course. APBIO Lohia. It may be, Angel. Looks like though you could be taking an AP exam that day. I mean, you don't have to get ready for an award night. Well, I guess you probably would want to. Sounds like you better get the hairs together then before that day. But I'm interested with COVID, if it's going to be more like last year. So if it's like, it's, you know, it's so hard to say because I don't know if kids are going to be vaccinated by then. If they do it like last year, it's all done virtually. So you still can get honored and get awards, but again, it's all virtual. The other thing I could see them doing is something similar to what they did for like graduation or other parts of that, where you are scheduled at certain times to come if you're being given certain awards. That way it limits the quantity that are there at once. I have no idea, Angel. Do Vale and Sale give that would definitely, Fadl, make for a more entertaining graduation. Normally, right, what happens is valedictorian will give a speech. We will likely have a speech from Mr. Galdes, Mr. Rudy, from some random human none of us know. It tends to be an older white male. Class president does speak. Yeah, superintendent is Mr. Galdes. I do know, I think the guest speaker this year is it's an old student that went here. I wish it was just the valedictorian and like class president. Just stop it there. Keep it going. Lots of speeches. But again, at that time in the school year, if not everyone is vaccinated, they might do it like they did it last year. And actually, Eli, there you go. No one wants to sit and listen to that. Sorry, I didn't say that on recording. Yes, I did. Last year's graduation got so much thanks and appreciation. Parents and students, I think, liked it more last year. So if you helped out or came to graduation last year, what happened was they recorded all the speeches ahead of time. So everything was recorded and it was all digital again. But they assigned kids to come alphabetically by their advisory at different times of the day. So like if you had me for advisory, you would come at 10 a.m. My kids would line up. You still walked across the courtyard. You still got your diploma from Mr. Galdes and Mr. Rudy. They still video recorded you walking the whole thing. But what was kind of, I thought, better and different is that anyone that you gave tickets to, they actually walked with you through the courtyard. So like if your family comes with you, like family would be cheering the whole time the kid was like walking through. Some had signs. Sometimes it was like an older sibling or best friend from another grade that was able to walk with them, which is unique, right? Normally that doesn't happen. So kids and parents actually really liked it. A lot of them said they felt like it was more personal and they were able to celebrate it more than if it was a traditional ceremony. I really liked it. It did take longer because of that, but you won't experience it longer. People helping experience it longer. I also got a wicked sunburn that day. Wow, the tangent, this conversation, just yikes. So you didn't get to celebrate with your class, but you did get to with your family. And that's a part that's not normally right. Normally they try to discourage people from like shouting and clapping during the ceremony, and your parents can't actually like go up there with you. But I would also say, like, if you're not vaccinated, do you want to celebrate with your class? Say no to COVID. How many tickets do you get? What is it normally, like 6 or something? 6 if we're outside, and then it's like 2 if we're in the auditorium. I feel like that sounds about accurate. Yeah. Valentina, you asked a while ago and I just totally ignored it. I'm so sorry. What kind of speech do valedictorians normally give? So what kind of speech do students normally give when they're graduating? I feel like it normally starts with some quote. They like kind of in a thesis statement, if you will, reflect on their time in the past 4 years here at Thurston. What a crazy trip it's been. Then they move into like things they learned from their experiences here in high school, how Thurston helped them grow. And then they wrap it up with like some kind of message of like, you're all awesome, we're all awesome, class of 2021, woo! So I know what one kid did one year, to give an example. Was this last year? Question mark. What is each year? It's a blur. Someone mentioned like the things they would miss from Thurston, and they were just the silliest things. So it, like, for example, like I'll miss the rotten burnt smell of Mr. Boylan's coffee coming out of his classroom. I'll miss hearing Miss Rosema scream down the hallway as she teaches her classes. Like, do you know what I mean? Like stuff like that as kids, they're like funny moments for you in any school day that other kids can relate to. So that was kind of funny. And you know, we've been done talking about the APBIO. So if you need to go check out, we are done with class in 2 minutes. If you have questions, email me. If you would like to stay for a couple more minutes, you're welcome to. I'm not kicking you out. Did my students ever show up? You mean the ones that stayed in here? Did they show up to their other class? So, oh, in my classroom, Lo Siento. Okay, they did not. So I think they're gonna be showing up in a couple, well, I don't even know, to be honest. I don't know. I do know, right, we were supposed to this week have between 44 and 60 kids come back to the building, and only 17 did. Now, some of that group is coming back in a couple weeks. Some of them also, I think once they realized, Dantes, you're good. Once they realized what it actually was compared to what they thought it was, they chose not to come back. So I do think our numbers are going to be significantly smaller than originally anticipated, which I'm fine with.